All right. Um, so I hope everybody had a good break. Um, the there's a problem set due on Friday by the end of the day, and it is posted on Moodle. I believe I emailed you guys about that. Also, remember that you have the adaptive immunity lab questions. Many of you guys already have completed them. And some of you guys have in fact already turned them in. But if you still have those, give them to me by lab. This week in lab, we're going to be working on doing the lab practical, which again is open notebook. So have your notebook ready for use then. And I also sent a doodle poll email right before this to schedule a doodle poll for the final exam review session, because the final exam is a week from today. And perhaps the third time is a charm in terms of pulling off the doodle poll. So today we're going to talk about transplantation. Just really briefly, remember that we've been talking about a number of different immune-mediated diseases, and we've divided them into kind of 4 different mechanisms. We were talking about these mechanisms with regard to allergy before break, and we spent a bunch of time talking about a type 1 hypersensitivity response, which was IgE mediated, a type 2 hypersensitivity response, which was mediated by IgG or IgM binding to a cell surface antigen, a type 3 hypersensitivity response, which is mediated by IgG or IgM binding to a soluble antigen, and a type 4 hypersensitivity response was sort of where I left us off. I'm not going to say, so much novel about a type 4 hypersensitivity response right now, but I was kind of mentioning it right at the end in a rush last time, so just to sort of be clear. A type 4 hypersensitivity response is different than the other 3 types in that the main effector, the thing that's really causing the issue in a type 4 hypersensitivity response, is a T cell, which you can see the T cells in blue in this figure compared to antibodies in all the other ones. Type 4 hypersensitivity response is also known as a delayed type hypersensitivity response. You can abbreviate that DTH, and so I may refer to it as a DTH. And the reason here is that, again, you are getting this response about 72 hours after exposure to the antigen. And so that's kind of a key piece to it. We talked about a type 1 hypersensitivity response, and we referred to it as an immediate type hypersensitivity response. But I told you that that was sort of with a caveat, because it was only immediate the second time you saw the antigen or later. Because the first time you saw the antigen, you needed to have sensitization and to have kind of a switch of immune responses, sort of activation of naive T cells. Say if we were talking about a type 1, switch to IgE production, all that kind of stuff. With a DTH response, it's the same kind of thing. We have 2 phases. We have a phase that happens the first time you see the antigen, which is the sensitization phase. And really that is kind of the same idea as the sensitization phase that we saw in other places, in that the sensitization phase is where we are actually activating naive cells. And so we don't see a response the first time we see the antigen. Instead, we see the sensitization phase. Specifically in the case of a DTH response, we are sensitizing a naive T cell to make that naive T cell become a Th1 cell and to have Th1 effector functions like making interferon gamma. And so in the sensitization phase, we are basically pushing T cells that respond to the antigen towards being Th1 cells. Sometimes when I think about a DTH response, I kind of think about that's just a fancy word for Th1s. And then the idea is that later on, when that T cell has already become a Th1 cell, if you see the antigen again, now that cell is going to be an activated Th1 cell. It's going to relatively quickly be able to start secreting cytokines, activating macrophages, and causing some amount of damage. You can see some parts of that DTH response here. Again, note that this is saying it's taking about 24 to 72 hours because the antigen needs to get into the body, needs to be presented. We need to have T cells get activated. Most likely, we may have to have some trafficking to the lymph node. We're going to have some T cells proliferating. See how originally there were a couple and now there's a lot more than a couple? All of those things are taking that time up to the 72 hours. Now we can actually start getting inflammation and changes in that location. There are lots of famous examples of a DTH response. One of many that's out there is the poison oak response. So if you are exposed to poison oak, actually there is a lipid, there's a compound in the poison oak called erythritol that actually reacts with a self protein, changes it to a new protein, and now it's a foreign antigen. And if you think about it, if you go out and get into a bunch of poison ivy or poison oak, you don't tend to have a rash like 10 minutes later, as you would have with a type 1 hypersensitivity response. It's a couple days later, you tend to suddenly have a rash. And that's because that is sort of this classic DTH response. The first time you were ever responding to poison ivy or poison oak, you were sensitizing T cells. The second time, you're getting this type 4 hypersensitivity response acting in that part of your skin. So I just wanted to give you that sort of brief reminder before I switch gears into talking about transplantation today. So we're going to talk about a few different issues regarding transplantation. This image shows a number of different organs that are commonly organs that are transplanted. And you can see on the left a number of different organs and how many transplants of those organs there were in 2016. So you can see there are things like heart, kidney, pancreas, intestine, liver, lung. This doesn't show cornea, but cornea would be another example. And so you can see it's a decent number. I will generally be using kidney as my example, partially because kidney transplantation is so common. One reason, of course, why kidney transplantation is really common is that you have 2 kidneys. You only have one of everything else. So you can be a living kidney donor by giving someone one of your kidneys while you keep the other one. You can't really give someone your heart and still be alive. Organs that are shown here. There's one other kind of general distinction that immunologists make, and so I want to kind of make that distinction really clear here. One type of transplant that immunologists often talk about is known as solid organ transplant. And when we're talking about solid organ transplant, we're really talking about the things on the left. The things on the right, the blood, bone marrow, are actually transplants of part of the hematopoietic system. And there are some rules that are unique to hematopoietic transplantation compared to solid organ transplantation. And so most of the time today, I'm going to talk about details about solid organ transplant. And then at the end, I'll talk about some of the things that are unique about hematopoietic transplantation. I want us to think for a second about kind of the transplant scenario. So one reason why a patient needs a transplant, of course, is because they are quite ill and have some other kind of serious issue, and without a new organ, they are probably not going to be able to survive long term. So that's a pretty serious situation. And typically, what has to happen when there is a transplant is that the organ that is transplanted needs to be transplanted very quickly, because that organ needs to remain viable. We need to not have a lot of cell death in that organ. We need to have that organ be incredibly fresh and really not have lost blood flow. And so generally what will happen is sort of almost immediately after an organ becomes available, that patient may be called up in the middle of the night, say it's the middle of the night, that organ is going to be, flown to the place where we have the recipient, and we're going to do that operation sort of right away, because this is such a severe situation for our transplant recipient. And their health. And there are a number of situations where the immune system can actually attack that organ that we have just worked so hard and gone through a huge amount of effort to transplant. The immune system can basically destroy that organ pretty quickly. And so it's important for us to understand this process to really be able to help out these patients. Also note that when I was talking about this, I mentioned the recipients. I am going to frequently mention recipients or donors a lot, so make sure you're paying attention to whether we're talking about a recipient or a donor in different situations. Originally, a lot of our understanding of transplantation immunology was figured out by doing skin transplants on mice, because mice are sort of readily available and are used in immunology for all the reasons we've talked about before, and it's pretty easy to transplant skin among different mice. And so if we were to take a recipient mouse who is strain B and give it a skin graft from a mouse of strain A, so some other mouse, we see that rejection happens in a couple of weeks. If we give that mouse another piece of skin from the same donor, from strain A again, so notice that we were giving a strain A graft both times, it's a strain B mouse. Now that mouse is going to reject that skin graft even quicker, in 6 days instead of in 14 days. And if you just look at that phenomenon, you can see, this looks like it might involve an adaptive immune response, because you're seeing this improvement in the response over time. The response is getting faster as the mouse sees the antigen again. And so this makes us start to think something's going on here with an adaptive immune response. What you can also do is take this mouse who has rejected this skin, and you can just take T cells from this mouse, give these T cells to another mouse. Again, a mouse of strain B, give that mouse a strain A skin graft, and you will again see that quicker 6-day rejection. And so this starts to make us think, there's something going on with T cells probably here. Because if we transfer just T cells can mediate this quicker rejection response. And so this starts to tell us there's probably something going on with adaptive immunity, and in particular, T cells might be particularly important. Brenna, did you have a question?
I just want to make sure the splenic T cells were from the donor.
The splenic T cells are from the recipient, this first recipient. So there was the
first rejection,
and then this recipient becomes a T cell donor. No, to its another mouse of the same strain. So this mouse down here is receiving two things. It's receiving splenic T cells from the mouse who's already rejected a transplant, and it's receiving skin. We now know that there are 3 different ways that transplant rejection can happen, that happen via 3 different mechanisms, although you will see as I talk about them today, one of them really dominates. And the one of them, oh my gosh, is the T cell one. I just showed you here. T cells are super important. It's because the T cell one is so particularly dominant. And so just like we've had those same types of responses that can mediate different types of immune-mediated disease, we had these 3 different mechanisms, and we saw them in allergy, we saw them in autoimmunity. We actually see the same 3 responses happening in transplantation. In transplantation, we tend to classify these 3 responses in terms of how long they take. So how long after the recipient has received their transplant does it take for that rejection to happen? And so that's the order that I'm going to tell you about them in. And so the first one that will happen is known as hyperacute rejection. This happens really quickly. Generally, with hyperacute rejection, we're talking about days following that transplant. And as you can see here, hyperacute rejection is really similar to a type 2 hypersensitivity response. So it also is going to involve antibodies, and it's going to involve antibodies against cell surface antigens. And so what this might tell you, and so one of the things that's really key about hyperacute rejection, I guess, is that hyperacute rejection actually involves pre-existing antibodies. So one of the things that's important about hyperacute rejection is that you already have antibodies to the foreign antigen before we do the transplant. That's why this can happen in days, because you already made these antibodies, you already have them at high level in the body. And so you can see here some pre-existing antibodies. When the organ, like the kidney that's shown here, is transplanted and gets connected to the bloodstream, those antibodies are going to go into it. And those antibodies might do things like start to bind to antigens that are on the cell surface, like the wall of capillaries, and actually destroy the capillaries in the kidney. Or allow neutrophils to inflame and destroy the kidney, or you can see some other destruction here. And so, of course, our patient sadly ends up with a kidney that looks like this, that has been destroyed. And so one of the things, again, that's really key with hyperacute rejection is that you've got to have pre-existing antibodies, because you don't have enough time to actually start an antibody response, activate the B cells, class switch, all that business, get memory cells, get plasma cells, all that stuff, to have this response. If you think about it, this is sort of curious. Do you think that you generally have antibodies against other people right now? It seems weird, right? And if you think evolutionarily, you could be like, I don't think I have evolved in response to other people's kidneys. This doesn't seem like there's something that probably there's a lot of evolutionary pressure for. Why in the heck do we have pre-existing antibodies that are going to really cause a problem here? And you can see these pre-existing antibodies causing issues here. No, those antibodies are not to scale in that person. If your antibodies were like as big as that, that would be bad. So here's your pre-existing antibodies, put it in the new kidney. The antibodies are going to destroy the blood vessels in the kidney. Kidney is going to be dead. Patient is going to die. It turns out that by and large, there's one antigen that tends to be the problem with hyperacute rejection. So there is one very frequent antigen that you do have antibodies against that is in other people. That is the blood group antigen, the ABO blood group antigen that I told you guys about before. So remember, for example, I have type B blood. That means I have antibodies against the A antigen because of similarities between those carbohydrates and some microbial carbohydrates. And so if I were to get a transplant from someone with type A blood, there would be type A antigen all over their cells. The antibodies that I already have against A antigen would destroy that graft, which is the name of the thing that gets transplanted. Destroy my graft. So if you were to think about it, if you were going to be the physician in charge of giving me a transplant, could you imagine a way that you could avoid hyperacute rejection? Yeah, Jonah.
Give you a kidney from someone with the same blood group as you?
Yeah, so basically you're going to do blood type matching before you do any transplant. And if you do blood type matching, then hyperacute rejection is not going to happen. We see hyperacute rejection very rarely now because we know to do blood typing beforehand. In fact, it takes maybe 5 minutes to do a blood type. Before every transplant, they're always done. Generally, if there is a case of hyperacute rejection, there is generally a very large lawsuit for the hospital system shortly thereafter, because it's pretty easy for us to get around hyperacute rejection. So we don't see hyperacute rejection very frequently. The big type of transplant rejection that we're going to spend most of our time talking about is the next quickest type of rejection, which is called acute rejection. And as you can see here, acute rejection is similar to the type 4 hypersensitivity response that I told you about earlier. When we think about acute rejection, we think about maybe a few months following the transplant in terms of where we start to see issues. And as you might guess from that description of acute rejection, where I said that it was similar to a type 4 hypersensitivity response. Acute rejection involves T cells. As you will see in a later slide, there are a couple of ways acute rejection can happen and can work. So this actually shows kind of one specific one. What you can notice is that in our kidney, we might have a cell. Here, they're listing a dendritic cell. That's the place where it's getting a little specific. We're just going to say a cell. It could be a kidney cell. It could be a dendritic, whatever. It's a cell from this kidney. That cell could at some point end up going to a secondary lymphoid organ. And that cell could activate some T cells. And so we could see some T cells getting activated, those T cells then going back into the kidney, and those T cells destroying the kidney. And at the bottom, we've got 2 or 3 different images showing T cells basically infiltrating kidneys. And so here you can see T cells that are going into a kidney and eventually destroying that kidney. And this is part of acute rejection. This is this process that we see. And you can see these sort of steps broken down here. Again, it's somewhat similar to what we've seen in the allergy situation. The difference is if I go get some poison ivy, which is also a type 4 T cell hypersensitivity response, how long is the poison ivy oil on my body? Like a day, a few hours maybe, and then it's gone. And then, so I have a phase of the response, and then it stops. And then I get the second phase if I ever see the antigen again. If I get a kidney, how long is the kidney in my body? For the rest of my life. And so kind of all of this is going to happen at once. There's not going to be like a time where the antigen goes away. And so it's the same set of phases that we saw, but they're going to be sort of smushed together. We're not going to think about them as having this big gap between them as much because the antigen is going to be there the whole time. And so again, you can see we've got cells that are in the kidney. Those cells that are in the kidney are going to head to a lymph node and are going to activate some T cells. And so that's sort of that first sensitization phase. But once those T cells are active, those T cells are going to leave the secondary lymphoid organ. They're going to head to the kidney, and they're going to start killing cells. They're going to start secreting cytokines, and they're going to destroy this kidney, which is going to be really sad for our donor. So some of the details of this were first worked out again in thinking about mice and thinking about mouse skin transplants. And you've actually seen pretty much these same experiments earlier in the semester, where these experimenters saw that if they took skin from a mouse of one strain, strain A, it's green, and gave that green skin to another strain A mouse, also green, then the graft was not rejected. You can see that is listed as a syngeneic graft. I would actually, there's a better piece of chalk. I would call it an autogeneic graft. And that's basically a transplant to yourself or to someone who's genetically identical, which in the case of mice, all the mice of a particular strain are genetically identical. They also show an allogeneic graft here at the bottom, where you can see we've got this mouse who's of strain B and is orange. We take a piece of skin from this mouse, we transplant it into the green mouse, strain A mouse, and you can see that that graft is rejected. And so this is an allogeneic graft. This is a transplant to another genetically not identical member of your species. So if you were going to get a transplant from another human, that would be an allogeneic transplant. And you would see rejection, as was seen in this situation. Yep, Juliana.
So on the other side where it said like
Mm-hmm. Yeah. Yeah. Yes.
Is this why when you can like give a skin graft to yourself, they go to like relatives?
So it's likely going to be relatives, but in fact, what they really want to do, the number one goal is to give a skin graft to yourself. The number one goal is if you're say a burn victim, is to take skin from another part of your body, because then it's an autograft, not an allograft, and you don't really have to worry about rejection in the same way. As you can see here. Well, if you actually were getting skin from somebody else, it'd be a bit more of a problem. And so what these immunologists did is they started doing genetics studies to try to find out about the genes that were important for this process. So they started doing different kinds of crosses to kind of find out what genes were really important for this. And this is actually how they discovered the MHC genes in mice. It was the major histocompatibility, whether or not tissues are compatible, histocompatibility complex. This is how they found MHC in mice. And so they did things like have the strain B mouse who was a parent. Here you can see the child. It has green and orange stripes because it's the child of the green mouse and the orange mouse. You can see the orange mouse could transplant while the F1 child could not transplant into the parent. And they realized it was sort of a question of whether or not you had the same MHC. So here, this mouse has orange MHC, so it can take orange skin. This mouse doesn't have both green and orange MHC, so it cannot accept the skin. And so these experiments said to them, okay, there's something, this is all about MHC. MHC is where this comes into. And again, you will notice I told you that's where the name comes from of MHC, the major histocompatibility complex. Histocompatibility is basically tissue compatibility. And so this is showing some data about kidney grafts and looking at kidney graft transplants and how long they survive in a patient. And this is looking at 10-year survival rate. So what percent of the transplants survive for 10 years, and then what's the half-life of the transplant. And what you can see, and again, it's showing us number of mismatches in MHC between 0 and 6. Because we're thinking about kidneys here, we're generally thinking about MHC class 1. And so you can see if the MHC is matched perfectly, there are no mismatches, we have the transplant last much better than if we have some mismatched MHC. And this is where I get to tell you that I oversimplified something that I told you about earlier in this semester. And so we talked about MHC restriction, if you recall. We talked about this idea of how T cells recognize something to respond against. And we talked about the fact that T cells are actually recognizing this combo of MHC plus peptide. We saw that T cells had to recognize both the correct MHC molecule and the correct peptide in order for that T cell to work, which could mean destroying stuff, like destroying the kidney. If the MHC is wrong, T cell doesn't work. If the peptide is wrong, T cell doesn't work, is how we've talked about this before. And we talked about the Zinkernagel and Doherty experiment, where that really led to figures like this in different textbooks, which was a great experiment, won the Nobel Prize. Yay. But what we really also understand is that if we think about MHC plus peptide, this is maybe a little more realistic way of thinking about what the T cell sees. And so this is a 3D model of the atoms in the MHC and in the peptide. MHC atoms are colored here in blue and white, while peptide atoms are in orange. Know that, of course, if you were the T cell, the atoms would not be different colors. And so if you look at this and you were to imagine this as a T cell who is colorblind, you would see that this is really one big surface of a protein. You know, it's not like an easy thing to think of two different checkboxes of what's going on with the peptide, what's going on with the MHC. And we often think about recognition as looking like what's shown here on the left, where we've got the MHC, we've got the peptide, T cells making contacts with sort of both of them. And alternatively, you know, we have a different MHC. So here you can see a different MHC. The structure of that other MHC is just so different. Look, this one's like flat and this one's like curvy. That the T cell definitely is not going to be able to respond to both of those things. They're just like so structurally different MHC-wise. All MHCs aren't quite that different. They're not quite as different as all this. It turns out, we now know, Zinkernagel and Doherty picked two mouse strains with MHCs that were like perfectly structurally different, so they got totally clean data. And if they had picked different MHC types, they would have had much messier data. Because sometimes we can have a situation where if we have this foreign MHC, so this is so here's what the MHC should look like in this green. Here's this foreign MHC. Maybe the foreign MHC has some amino acids that actually make contact with the T cell receptor, where in this situation, peptide ones would. So maybe, as far as this T cell is concerned, this foreign MHC is this MHC plus peptide. It can recognize this foreign thing as think that it's this MHC plus peptide, because the structures just so happen to look that way. You could also imagine that maybe it could look like this, where a combination of a particular peptide and a particular MHC might kind of fool the T cell into thinking it's seeing this self MHC plus self peptide. And so sometimes we can see situations where foreign MHC kind of looks like self-MHC plus foreign peptide. And so we can get what's known as allorecognition. And so the issue here is sometimes somebody else's MHC might look a bit like your MHC with foreign peptides on it. And so your T cells are potentially going to get sort of confused. There are two ways we think about allorecognition to happen. I've actually kind of been describing one of them right now. And I've kind of been focusing on that one because that's the one that like people's brains have a slightly harder time with. I think the other one's a little more straightforward. But know that there's sort of two things going on here. I've really only been describing one. And these two things are known as direct and indirect allorecognition. And so this slide, as well as the next slide, are going to talk about direct versus indirect allorecognition. I'm going to talk about direct on this slide and the next slide, and I'm going to come back to this and do indirect on both. And so direct allorecognition is basically what I've been telling you about previously, where you can see here is my antigen presenting cell from the donor. So this is some cell from the kidney of the donor. It's presenting on the donor's MHC, whatever MHC that kidney donor has. And the recipient's T cells are getting sort of fooled. They think the donor's MHC is self-MHC plus peptide. And they're like, oh yeah, that's a bad thing. I got to kill that. So here you can see that the sort of donor MHC is just directly recognized on antigen presenting cell from the donor. You can also see this on the left here. And so here you can see, okay, we've got some cell from the donor. It could be a dendritic cell that happened to be in the kidney when we took it out of the person. Could be a kidney cell, whatever. I don't care right now. It's going to be presenting stuff, and that stuff is going to look foreign to the recipient's T cells. And so we're going to turn on those T cells in that way. The other way that we can see allorecognition is through a process known as indirect allorecognition. In order to understand indirect allorecognition, we have to think very briefly about one other kind of biochemical piece of information. Don't worry, it's not hard. What kind of biomolecule is MHC? Yes, David. A protein. So if you think about it, if I transplant a kidney into you, and that kidney has MHC on its surface, and that's an MHC that's different than your MHC, that's a foreign protein as far as your body is concerned, right? It could happen that that foreign protein could get broken down and put on your MHC. So you could take that foreign MHC, break it down, and present it on your MHC as a foreign protein. And so here we are recognizing the donor MHC on the recipient's cells. So the donor MHC is getting broken down into a peptide, and it is the peptide getting presented by the recipient cell. And you can also see that here. So what I want you to notice in this image is that donor is yellow. So that's really obvious in the one on the left. Here's the one on the right. You can see the donor MHC is yellow. There are a few situations where we can imagine this happening, but I'm just going to kind of give us one scenario. So again, I want you to imagine this kidney transplant happening. We're going to, take a kidney from one person, put it in another person. That kidney is going to be not connected to a blood supply for a little while. It's going to be outside of the body for a little while. Do you think every single cell in that kidney is going to survive the transplant process of getting like cut out of one person, being not attached to a blood supply, being sewn into another person? No, probably not. There are probably going to be some dead cells in that kidney, right? And so we might have some of those kidney cells when we put them into the recipient that are dead. And those dead cells will have MHC, in this case, the yellow MHC. Those dead cells are going to get phagocytosed and cleaned up by the recipient's body, just like any other dead cell gets phagocytosed and cleaned up. And so now you can see, here's phagocytosis of that dead cell. And now when that is phagocytosed, we're going to degrade all of the proteins that were there. You can see that means even the MHC gets degraded. And so now what was yellow MHC became little yellow peptides. And that yellow MHC could be presented on the recipient's MHC. So the orange MHC might present one of these yellow MHC peptides and turn on a T cell. And so this would be indirect allorecognition. And so these are the two ways that mismatched MHC can lead to activating T cells, which will result in acute rejection. If you look at the data that I'm showing you here, there are a couple of questions that sometimes people have. And the question that I want us to think about right now is about this zero mismatch situation. So if we have zero mismatches, you can see that it's not 100% of the transplants stay. It's not like the transplant stays forever. And so there's something going on besides MHC here. And so we need to think a little bit about what's going on with zero MHC mismatches. This is the short answer, but I'm going to explain to you what it is. And the thing is that it turns out there's something in addition to the major histocompatibility antigen. There are also a group of antigens called minor histocompatibility antigens. And this slide describes minor histocompatibility antigens. And you might think about it and you might say, okay, like major histocompatibility complex, MHC, that was kind of a whole complicated business. So minor histocompatibility complex, that's going to be more complicated stuff. Oof. But actually, it's not. Minor histocompatibility complex is like a fancy word for something really simple. So I'm going to give you a sort of simplified example right now, and then we can think about other examples, and you'll be like, oh, that's all this is. So let's imagine that I desperately need a kidney. I know it's very sad. We find that David and I have a perfect MHC match. 100%, all of our MHC class 1s, all of our MHC class 2s, perfect MHC match between me and David. So David is going to give me a kidney. Can you think of any proteins that might be different between me and David? Are there any proteins that David might have that I don't have? And so people are shaking I see some yes head shakes. Can anybody give me any examples? I did use David on purpose. Yeah.
Proteins encoded on the Y chromosome?
David is male. So David has that Y chromosome, and he has a whole bunch of proteins that are encoded on the Y chromosome. My body has not seen any of those. Those are all foreign proteins to me because I don't have a Y chromosome, right? All of those are foreign as far as my body's concerned. Those would all be examples of minor histocompatibility antigens. Minor histocompatibility antigens are any gene that varies between two people. It's possible that, for example, I get a kidney from Brooke. Well, Brooke has some protein that allowed her to have blonde hair, and I have a different protein, and so my body responds to the blonde hair protein because it's a difference between two individuals. So minor histocompatibility antigens are basically any other genetic difference between two individuals of the same species. Again, the Y chromosome ones are pretty famous. People do a lot of experiments with those. And so what you can see here is we have our donor. Our donor has the little flower self-protein. And in the donor, the flower self-protein has a red dot on it. In the recipient, that has a blue dot on it. That might be because of whatever kind of mutation happened between these two individuals. Blue eyes, green eyes, like whatever. Any genetic change you can imagine between two individuals. This is red dot versus blue dot. Well, that means that when this gets presented on MHC, the red peptide is shown, and here the blue peptide is shown. And so just because we have these two genetically different individuals, we're going to see some minor histocompatibility mismatch. And so basically, unless you are transplanting from yourself, there's a minor histocompatibility mismatch. There is no other way to make there not be a minor histocompatibility mismatch.
Twins?
Maybe twins. I could see argument. You could maybe twins, but it would be tough, and I could argue on that one. This is actually one reason why some parents, after they have a baby, actually save the baby's cord blood. So if the baby ever needs a blood transfusion, they can get blood from themselves, and things like that. Or why, in certain cases, there are a number of cases, bone marrow transplant as well, if you're going to have a treatment that's going to really mess up your immune system, you actually donate bone marrow to yourself before the treatment so that you can get your own bone marrow back. Because you can actually avoid minor histocompatibility mismatch if you're sort of getting transplant from yourself. And one thing I will say is that in general, if you have a mismatch in major histocompatibility, that graft is going to get rejected pretty rapidly. If the only mismatches are in minor histocompatibility, you will still see acute rejection, but it will be a little bit slower. Because of course that's not, MHC is on every single cell, and there's huge amounts of it. We do also have this other type of rejection, which is known as chronic rejection. Chronic rejection is seen years after transplantation. There's a lot that we don't understand about chronic rejection. And one of the big reasons why we don't totally understand chronic rejection is that chronic rejection was only something we could see when we kind of had a way to deal with acute rejection. Because your transplant has to live through months before it could possibly get rejected at years. And so only when kind of we figured out ways to deal with acute rejection, we figured out, there's all this other response that happens too, even longer away. And chronic rejection has a lot in common with a type 3 hypersensitivity response. The type 3 hypersensitivity response was immune complex disease. So that's where we had free antibodies and antigens that were forming immune complexes and blocking things like blood vessels and causing some kind of damage. And so you can see this here. With chronic rejection, we have immune complexes that get formed. So again, we're going to have antibodies that are formed. They're going to deposit in the vessel wall, perhaps the vessel wall of the kidney, and lead to inflammation and damage of that vessel wall, and lead to damage of the kidney. One thing that we do understand about chronic rejection is that these antibodies are not pre-existing antibodies. So in the case of hyperacute rejection, the antibodies were pre-existing. These are antibodies that you become sensitive to after the transplantation. You didn't have them before transplantation. So again, this is some perhaps, protein that differs from individual to individual, that you've somehow started making an antibody response. You probably had to also prime a T cell response. There are probably a whole lot of steps going on here. And this is going to happen sort of years after the transplant. And again, there's not as much known about this situation. There is one other issue that I need to tell you about, sort of general transplant rejection issues, which is if we think about solid organ transplantation versus bone marrow transplantation. And so in the case of bone marrow transplantation, there is an additional kind of complication. There is an additional bad thing that can happen with bone marrow transplantation. And so on the left, you can see a solid organ transplant, like with the kidney. So here we're going to transplant the kidney into this person. And so we're going to have a donor kidney going into the recipient. The recipient's T cells are going to try to kill the donor kidney. In a bone marrow transplant, sometimes you can actually you're putting bone marrow into a person, and the bone marrow can try to kill the rest of the body. And so the graft can try to kill the host. It's known as graft versus host disease, instead of the host trying to kill the graft. And so the bone marrow can actually cause an issue in bone marrow transplantation. So just to give you a brief sort of piece of information on this, these are some diseases that are treated by bone marrow transplantation. A number of them are genetic diseases where there are issues with immune genes, and so we want to give someone bone marrow that doesn't have that issue so that they will have an immune system. So for example, if you don't have RAG1 and RAG2, you can't make B cells and T cells. So we just give you some bone marrow that does have RAG1 and RAG2. And then you can make B cells and T cells, and you live happily ever after. Alternatively, cells of the immune system are generally rapidly dividing cells. And we can have a number of situations where we need to kill rapidly dividing cells because of cancers. And so for many types of cancers, we will actually use a bone marrow transplant after we have treated our patient. We may have to get that from others. We may sometimes be able to use our own bone marrow. But so the basic idea is, we have our patient. Our patient has this full immune system, but also this tumor that's dividing rapidly. We're going to use radiation and chemo. It's going to kill all the rapidly dividing cells. Yay, kills the tumor. Sad, also killed the entire immune system. See, now our patient is empty. And so the way that we give this person an immune system back is that we give them bone marrow. So we actually go into, you can see there's bone marrow aspirate there. We put bone marrow into that person and we basically refill them up with bone marrow. And so bone marrow transplantation is not like a weird kind of niche thing. There are a lot of situations where bone marrow transplantation is incredibly important. But the problem, like I said, with bone marrow transplantation is described here. The bone marrow as a location is a location for the immune system that has a lot of great cytokines, a lot of great stuff to like help developing B cells and developing neutrophils and developing stem cells, all sorts of things. It turns out that some of our memory cells, like our memory B cells, our memory T cells, also like to hang out in the bone marrow because they can get some cytokines there and they can get this nice, happy immunologic location. And so bone marrow is a mixture of cells. Here you can see this yellow stem cell. You can also see there are some memory T cells. There's a whole variety of cells in that bone marrow. And when we transplant this bone marrow, the stem cells are sort of the thing that we want to grow a new immune system for the person. But these memory T cells and memory B cells that happen to be living in the bone marrow will go into the patient. They will likely they'll go to the patient's lymph node because that's what B cells and T cells do. They'll be like, oh my gosh, look at all this foreign stuff. Everywhere I look, all I see is foreign. And they're going to get really activated in the patient's lymph node. And then they're going to actually start destroying all the tissues because all the tissues are foreign after you've transplanted in those memory T cells. And so you can see some examples of graft versus host disease here. There are different severity grades, but you can see that those severity grades can involve different amounts of your body surface being covered with a rash, different amounts of liver destruction with liver proteins being made, and different amounts of GI destruction. Look at that, and I want you to think about like sizes of liter bottles of how much diarrhea per day that is. Like, this is not good. This is a very, very severe situation. We had this patient who had a severe medical condition that required a bone marrow transplant, and this is the end result. It's like adding a lot of insult to injury here, right? But we have largely gotten rid of graft versus host disease. Or graft versus host disease happens way, 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 way more rarely now than it used to. So I almost feel weird like telling you about graft versus host disease at this point. Because as I described graft versus host disease here, I sort of was also saying some of the things that we've realized can be helpful and can make graft versus host disease go away. Because if you remember, I told you that the bone marrow is a mixed population of cells. It has both the stem cell we want and all of these other cells in it. So if you had to guess, how might you make sure that graft versus host disease doesn't happen? Yeah, Brenna? You isolate just the stem cells. And so generally that's what we do at this point, is that we'll take bone marrow and we'll find, you can see that it has stem cells, B cells, and T cells, and we'll treat that bone marrow to just get the stem cells, or the hematopoietic stem cells, HSCs, and then we'll do a transplant of just the HSCs, so we don't have to worry about this situation. Originally, so like when I was in college, there was a big push for bone marrow registry to get registered for your HLA types for matching, and people were always like, oh, I really want to help, but being a bone marrow donor sounds terrible. They have this big giant needle into your hip bone to get like marrow out of your hip bone, and that just sounds awful. We actually have realized that hematopoietic stem cells can circulate in the blood sometimes, and so we can actually pull it out of the blood at this point. So to be a donor, you actually just have to donate blood instead of having to have the giant needle in your hip. So this has all gotten so much better, so much easier. It's awesome. MHC matching is really important in transplantation and in bone marrow transplantation. As you guys correctly figured out earlier in the semester, if you can put in, like, you could put in all sorts of bone marrow and it would just learn what is self based on the thymus. But sometimes if you don't do matching, you end up with really weird scenarios where, like, you get T cells out of the thymus, but they can't recognize anything in the body. So you do have to have matching in bone marrow transplantation, but it's for different reasons. The other thing that's really important about transplants is yay cyclosporin. Transplantation was sort of rarely a thing. People didn't have transplants that survived very long until cyclosporin A was invented. And now that we have cyclosporin A, transplantation can sort of be a thing. And so because of cyclosporin A, transplantation has sort of been a big thing. I'm just going to tell you this one additional piece because it makes more sense to do it this way. Another really major problem with transplantation is the current transplant waiting list. So this is actually showing you the number of donors in yellow, the number of transplants that actually happen in blue, and the number of people who need a transplant in orange. And so right now there are way more people who need transplants than there are organs. And so one of the other things that a lot of immunologists are thinking about is how do we actually make more organs, or how do we get more organs, or how do we deal with more organs? There's one idea that some people have. There's some sort of biomedical engineering ideas. There's something called xenotransplantation, about transplanting organs from other species. There are a couple other species that are really interesting in this regard, which I can say a lot. One other thing that I'll just mention is that if you look at the countries have different policies on organ transplantation based on, do you have to opt in or opt out? So basically, if you die and you didn't tell them what you think about your organs, do they assume yes or do they assume no? So like in the US, if you don't tell anyone what you want to do with your organs, they assume no. And so in countries like that, you can see this is what % of people actually like decide to not be lazy and say, yes, to transplant my organs. It's like, between 10 and 20%. In some countries, they transplant everybody's organs unless the person actively refuses. And in that case, it's something like over 90% of people are donors. And really, that's just a matter of people's laziness. Like, what are you going to do with the lazy people? Are you going to, people who are too lazy to fill out the form, are you going to assume yes or are you going to assume no? And so even some sort of public policy things are a big part of it, but there are also ideas about biomedical engineering approaches, stem cell approaches, as well as animal transplantation. Pigs are, in fact, the number one organ or animal that people talk about because it turns out their organs are the same size as ours in terms of volumetrics. Right? Anyway, so that's the end.